Welcome everybody to the HEALEY ALS Platform Trial Weekly Update. Today we're really happy to be having a special presentation by our colleagues at Biohaven to tell you about one of the regimens, Regimen B, with their drug called Verdiperstat. I know many of you have come weekly, and I'm glad to see you back, but there's also some new people. So before we jump right into their presentation, I want to give you a little background about the platform trial. So first, I'll just say that this is a community effort to really accelerate how we develop treatments with people for people with ALS. And you can see from this slide the sheer number of foundations that have provided support, patients who have provided philanthropic support, and our industry partners and our colleagues at the Northeast ALS Consortium. Next slide, please. So the traditional way drugs were developed in ALS and for many diseases was really to test one drug at a time. We wanted to really change this paradigm to speed the process up because the number of drugs in development for ALS is growing exponentially. There's over 160 companies working on programs for ALS, and there are a lot of drugs that we're ready for bringing forward to patients. And we really felt that if we kept doing it one drug at a time, we would be here for many, many years, and we wanted to speed that up and really work on what all of you have really told us which is to work on the ALS clock. So we borrowed an idea already very successful in some cancers, in particular lung cancer and breast cancer, called a platform trial, where instead of testing one intervention at a time, we test multiple, and we share the infrastructure, and we share the placebo data, and we share some of the costs, and all that really brings down the time to getting answers and getting effective treatments. It brings down the cost, and it greatly increases for participants the chance of being an active drug. So we thought that this was the right time to bring this innovation to ALS, and we're very excited that this launched enrollment last summer, even throughout the pandemic. Next slide, please. So we are now testing 4 different treatments in this regimen. For a particular person who wants to be part of the study, you come in and you're randomized to one of the 4 available drugs. And today we're going to hear about Regimen B, Dippostat. And then you'll know which one you're on, whether you're on Regimen A, B, C, or D. But then there's another randomization to active drug or placebo, with 75% of people getting active drug and 25% placebo. And that's for 24 weeks. And after that 24 weeks, let's say you are in the Regimen B arm, you would have the option to go into what's called an open label extension. And that's a period of time where you get the active drug for sure, 100% of people do, and you can continue on that until we know the results of the blinded period, 24-week period. And that can be for some people 6 months, some people 1 month. It really depends on where the trial is when you enroll in that study. So the efficiencies here, again, are the shared placebo, the shorter duration of a trial than used in the past, 6 months. We do think that's the minimum time you need to be able to tell if a drug works or not. And then the open label extension for participants. Next slide, please. So for each of our regimens, we have 2 academic leads, and we're very excited about this for many reasons. One is there's a lot of work, and we need all the bright brains helping us, but it's also an opportunity to teach new investigators in ALS how to lead multi-center trials, because we need to keep attracting the smartest and the best people to our field to help us solve this serious illness. So for the Verdiperstat regimen, Dr. Jinsy Andrews from Columbia University is the regimen lead, and Dr. Suma Babu from Mass General Hospital is the regimen co-lead, and they've both joined us here on this webinar, and will be part of the Q&A after we hear from Biohaven on their drug mechanism of action. Next slide, please. Just to remind you that these webinars happen weekly. This idea came from our patient advisory committee. We record all the webinars, and they're posted up on our website, and we will be recording today's webinar as well. Next slide, please. And with that, I would, with much pleasure, turn the Zoom podium over to Dr. Irfan Qureshi from Biohaven to tell you about their drug.
Thank you so much, Merritt. It's a pleasure to be here, and thank you to the audience for listening to our presentation about regimen B for Verdiperstat. So next slide, please. Verdiperstat targets an enzyme which is called myeloperoxidase, or MPO. And what is MPO? MPO is an enzyme that is a critical enzyme in the immune system, and it's very abundant in myeloid cells. That's how it gets its name, myeloperoxidase. And myeloid cells are immune system cells like white blood cells. And in the nervous system, the immune cells that there is a lot of MPO in are microglial cells. And these are immune or housekeeping type cells in the brain. And MPO typically in a healthy person would end up attacking bacteria and viruses, but in a disease state, it can actually contribute to disease processes like oxidative stress, inflammation, and cellular injury. And in ALS, we know microglial activation is really a contributor to these types of processes that lead to the disease. And there's a lot of evidence that MPO is involved in many different neurodegenerative diseases, including Parkinson's disease, including a disease called multiple system atrophy, as well as other diseases. And it includes ALS. Next slide, please. So how does regimen B's study drug Verdiperstat work? As I said, activated microglial cells in the brain, they express the MPO enzyme. MPO produces the toxic compounds that can damage the cells. And Verdiperstat is an enzyme inhibitor, and it binds to and inhibits the myeloperoxidase, making it inactive so that it doesn't produce toxic compounds. And we believe that Verdiperstat will reduce oxidative stress, neuroinflammation, and protect neuronal cells from dying in ALS. In the cartoon here on the left side, in the healthy side, you can see microglial cells that promote homeostasis or healthy brain. And on the right side of the cartoon in ALS and other neurodegenerative diseases, you can see the microglial cells are activated and they secrete factors, including immune factors and oxidative and nitrosative stress factors that can lead to neurodegeneration. Next slide, please. So how does Verdiperstat work? This is just a simple diagram that shows you what I just told you. Verdiperstat blocks the MPO enzyme activity, and that is intimately linked to the microglial activation, neuroinflammation, and oxidative stress. And by inhibiting MPO, Verdiperstat, we hope, will reduce neurodegeneration. And Verdiperstat is first in class. It's the first kind of MPO inhibitor that's been studied. It's very potent. It's brain permeable, so it gets into the brain. It's very selective for the MPO enzyme, and it's orally administered, so it's administered as a tablet. Next slide, please. So I've told you that Verdiperstat reduces microglial activation. What is the evidence for that? Here, this is an example of some of the animal model data that we have available, and it's from an animal model of multiple system atrophy, which is a type of neurodegenerative disease. And in this slide, the panels A, B, C, and D show many different facets of the potential benefits of Verdiperstat. So in panel A, we can see that the dose-dependent reduction in microglial activation. So this is really what we are trying to achieve in the treatment of patients with ALS, is to reduce microglial activation. In this model, we see that we're able to do that. And as a sort of sequelae of those reductions in microglial activation, we see a reduction in the type of pathology that's found in MSA. We see a reduction in neurodegeneration, and we see an improvement in motor function. So these are a lot of beneficial effects that are seen in animal models of a neurodegenerative disease. We have similar data in other animal models, such as animal models of Parkinson's disease. Next slide, please. So I talked a little bit about animal models, but what about people? This slide shows evidence that Verdiperstat reduces microglial activation in human beings. So this image here, or these images on these slides, are a special kind of PET imaging or brain imaging that was done in patients who have Parkinson's disease and were treated with Verdiperstat. And when you look on the right-hand side in the panel, that is the PET imaging of one person who was treated with Verdiperstat. And you can see in the baseline scan, there's a lot of yellow and red, and that yellow and red is very indicative of inflammation and microglial activation. And you can see after several weeks of treatment that there is a very significant reduction in the levels of microglial activation and neuroinflammation that are associated with Parkinson's disease, just like they are with ALS. And on the left-hand side of the slide, you can see this is more of a graphic that shows the means amongst many different patients, not just a single patient. And it shows the PET signal is significantly reduced in many different Parkinson's disease-related brain regions that have microglial activation. So these are really remarkable results, and we're very excited about them. And they're some of the basis for why we think Verdiperstat could have a similar beneficial effect in people who have ALS. Next slide, please. So I talked about multiple system atrophy. I talked about Parkinson's disease. What is the rationale we have for using Verdiperstat in the HEALEY platform trial? And why do we think it may help people who have ALS? So Verdiperstat targets mechanisms, as I said, like oxidative stress, which we know is very important in ALS, microglial activation, and neuroinflammation. We know that human ALS patients have microglial activation, and you can see that using PET imaging, the same kind of PET imaging that I just showed you in Parkinson's patients. And I'll show you in a couple slides as well in people with ALS. And Verdiperstat is really the only compound in development that has shown the ability to reduce the PET signal with this very specialized tracer for microglial activation called PBR28. And so we believe that Verdiperstat is very uniquely positioned to treat people who have ALS. Next slide, please. So here are those images I just mentioned. The evidence for microglial activation in ALS comes from many different sources. These are some images from PET imaging from a paper that was published by our colleagues at Mass General, which shows, I think, really interesting data. The first panel at the top shows that there is an increase in microglial activation in ALS patients compared to controls. The second panel shows that the microglial activation co-localizes in the areas where we know the disease has an impact, where the cortex, the motor cortex, and related areas are impacted. And in the last panel, I know it's a little hard to read, there are correlations between measures of clinical function, things like the ALS functional rating scale, which is used to assess ALS in the clinic, and microglial activation. So these are really interesting data, which suggests to us that reducing microglial activation in ALS with a drug like Verdiperstat could be very beneficial. Next slide, please. So I talked a lot about biomarkers, about things like microglial activation. What about, is there any evidence in patients, in people with Verdiperstat? And yes, we do have phase 2 data in multiple system atrophy, as I mentioned, which is a neurodegenerative disease like ALS. And this is some data from a very exciting phase 2 trial that was done with Verdiperstat. And this was a relatively short trial. It was a 12-week trial, and 2 doses of Verdiperstat were studied. So we have a placebo group and a 300 milligram twice a day of Verdiperstat group and a 600 milligrams twice a day group of Verdiperstat. And this study sort of very interestingly showed a trend towards reductions in disability in people with MSA over 12 weeks, which was pretty remarkable for a disease that progresses very rapidly, to see a potential impact with a small sample size over a short duration of time. And in MSA, Verdiperstat is currently being studied in a large phase 3 fully powered study that's a 1-year duration. Next slide, please. So in conclusion, we believe that there's a strong scientific foundation that suggests microglial activation is involved in ALS, and that the ability to reduce microglial activation with an MPO inhibitor like Verdiperstat, which was shown in patients with Parkinson's disease, and also the Verdiperstat potential benefits shown on clinical measures in multiple system atrophy, really support the hypothesis that Verdiperstat could have a beneficial impact in ALS. So with that, I think that's my last slide, and I appreciate your attention. Thank you.
Thank you, Irfan. I want to also introduce now Mary Donahue and Lila Brady from Biohaven, and they're here to answer questions too. And again, Jinsy Andrews and Suma Babu, the leads. So we have a couple questions coming in, so please type your questions in the Q&A, and we'll have lots of time to get to all of them. I'm going to start with the questions related to the Verdiperstat. So this is for Irfan and your team. Is Verdiperstat used in the treatment of Parkinson's? Is it able to effectively reduce progression? And are its effects and benefits long-lasting in those patients?
Yeah, so that is a wonderful question. And the simple answer is that data is not available. We are studying at Biohaven multiple system atrophy. We're sort of a small but growing company, and we really want to study Verdiperstat in many different diseases, but we started where there was a lot of evidence and a huge unmet need in multiple system atrophy. And we are now studying with our collaborators at the Healy Center the role in ALS. And we do hope to do studies in Parkinson's disease in the future as well.
Thank you. Jinsy, I was hoping you could answer these next 2 questions, which are really more about the platform trial or trials in general. One is whether you know which regimen you're on up front, or do you have to wait until after 24 weeks?
Yeah, I think we talked a little bit about this earlier, but you know, when a person comes in to a screening visit, the first visit is your screening visit for the master protocol, right? So there you sit with the physician, you sign the informed consent, and then you go through a process of testing to see if you're eligible for entry into the master platform. After that, you'll have a subsequent visit, and at that time, you'll be randomized to one of the available regimens. And when you sign that informed consent form, at that time, you will know what regimen you were assigned. So everyone who is assigned to Verdiperstat should have received an informed consent. Right on top, it says Verdiperstat. And so you will know upfront when you enter into the trial which regimen you were assigned. Now, within that, there's a randomization, and there's a small chance that you might be randomized to placebo or to whatever regimen you were selected, and that time you would be committing to is a 6-month period, after which you have a couple of options. And I just finished ALS clinic, and I had one person who was actually coming to the end from a different site, but had this difficult decision to decide whether to continue to roll over to what's called an open label extension, which is where the person who participated in the trial would know they're getting the drug, the sponsor knows they're getting the drug, and the physician caring for them knows that they're getting the actual drug. So that's the open label extension. So it's usually offered to people who complete the clinical trial. Or alternatively, if they're still eligible, they can choose to re-randomize into the master the platform trial and be re-randomized to a different regimen. So it is, I say it so easily, but the decision is a little bit complicated. And once I worked through with it just today with somebody trying to think through what would be the most ideal, because they were eligible to re-randomize or to choose the open label extension or choose something else. So it's good to have those options. I think it's a good problem to have.
Yeah, it's important to have those discussions with your neurologist or neurologist like Dr. Andrews, who can take the time to go through all the good options. So there's a few more Verdiperstat questions. I'll go back to those now. So Irfan, one is with Verdiperstat specifically and the HEALEY trial in general, is this arm and the other arms, are they considered a phase 3 trial?
So I think that it's really important question. It's certainly a trial that has been discussed with the FDA, and if the study design is supported by the FDA, then you could call it a phase 3 type trial where the study drug, if it works, can be approved. I don't know, Merit, do you want to add something to that?
Yeah, no, I agree. I mean, I think you know, is a push to get away from those titles of phase 1, 2, 3, where one is you know, one is kind of dose finding, exploratory, and 2 and 3 are more like looking at efficacy and could it be a registration trial, meaning a trial that, if positive and robust, you know, could lead to approval.
Yeah, so I agree. I think sometimes in rare diseases, phase 2 and 3 don't really have that much of a different meaning. And I think what's really important is that the HEALEY team has really discussed the trial with the FDA. The FDA has endorsed the trial. And if there is a benefit, the FDA is willing to look at that data and potentially the drug can be approved.
So there's a couple of questions about side effects. So what type of side effects have been seen that are associated with Verdiperstat?
So I think that probably the best thing is if you're interested in learning about Regiprostat, if you're talking to your doctor or your neurologist about the specifics of Verdiprostat and the potential risks and benefits, that's probably the most appropriate thing that you should do.
I'll go maybe back to some platform trial questions, and maybe I'll ask Jinsy this first, and then Suma, if you could weigh in too. So why do most trials only accept patients with symptom onset of 36 months or less? Most people with ALS take that long to get diagnosed. Is it because the results are more promising?
That's a tough question. And a good one, actually, because it is important to understand how we evaluate the effect of any intervention. And so some trials actually has had even shorter symptom onset time. So some trials even restricted it to 18 months, 2 years, but we're trying to be as liberal as possible. I think as a community, we've come together to try to expand the eligibility criteria to be as inclusive as possible. If we're observing a group of people over a short period of time, the other thing is we're trying to get to an answer as fast as possible. So instead of a year or 18 months, like we did back in the Riluzole days, we're doing this trial in 6 months. So in that duration, we're trying to find an effect of whatever drug that we're testing. And so usually if we include people that are a little bit longer from their onset, usually it implies that they might be a slower progressor. So it might be harder to see that effect in that shorter duration. But, you know, we're doing our best to try to expand it with each opportunity to try to expand that eligibility criteria and not be as restrictive as we have been previously.
Suma, would you like to add anything?
That is a great question, and Jinsy, completely agree with you. I would just add also as a second prong, in addition to thinking about trial design for platform trial, there is a big push to reduce the time from symptom onset to diagnosis, and that's important as well. And many patients take very long for their diagnosis, several months, sometimes up to a year. And if we can diagnose ALS early and provide access early to patients to both FDA-approved therapeutics as well as experimental therapeutics, that'll be another great step in the right direction to accelerate drug development for ALS.
So we have a question that's, again, kind of related to the one about phase of development, but maybe Irfan, if you and your team want to tackle this one first. It's my understanding that the study is a phase 2/3. Can you talk more about this blending phasing and any guidance about potential timing for FDA approval if there was another phase versus this being your final phase, or I guess versus this one trial versus needing another one?
Yeah, so this is a great question. And, I'll take a step back and say, as we said a little bit earlier, what you call it is less important than how the FDA views it. And the FDA doesn't always take a one-size-fits-all approach. And really what they're very interested in is making sure that the drug is safe and, very importantly, effective. And so sometimes the typical standard is 2 large phase 3 trials to demonstrate efficacy. But in rare diseases and in diseases where they've had a lot of interaction in developing the study, they may be willing to approve a drug based on a single study. So really, I think there are no one-size-fits-all answer. It will depend on the FDA, their view of the data. If the data is resoundingly positive, I think that this study is designed as a trial to support the approval. The timelines, again, there's no one-size-fits-all. There is the hope that if the drug is, any of the drugs in the platform trial really are effective, that the FDA will give an accelerated review and try to get them to people as soon as possible.
This is a platform level question that either Suma or Jinsy you want to answer. So understanding that these are phase 2/3 drugs, so adverse events should be limited. But suppose there is an adverse effect to an initial regimen a patient is on. Is there an opportunity to try a different one?
So Suma, you want to take that one?
Sure, Jinsy. Yeah. So certainly, there are a number of checkpoints built into the platform trial design. If somebody does not tolerate a certain medication, first we assess, how bad is the adverse reaction, and is it mild, moderate, or severe? If there is a point where a patient cannot continue forward on the study drug, there are options to continue the trial on an intent to treat method, which means that you're not on the drug, but you will still complete all the outcomes and complete all the 6 months of the placebo control. And then you'll have the opportunity to re-enter the original pool and be randomized to a different regimen. There is an option also to stop the trial and wait out a certain portion after stopping the medication, and again coming back to the screening phase at the master level and re-entering and be randomized to a different regimen.
Thank you. So there's a question of what, if any, supplements have to be stopped to be in the trial? Jinsy, do you want to answer that? Maybe big
Yeah.
picture.
Yeah. There are some requirements in most trials where you are not supposed to be taking any other investigational products while you enter a clinical trial to take another experimental drug. But typically, I think in seeing people in the clinic and screening them for the platform trial, the one supplement that I do often have to discuss is high dose, ultra high dose vitamin B12. If it's delivered intramuscularly, can be an issue, and that needs to be stopped prior to enrolling. And the other one is usually people are taking L-serine, and there is a clinical trial of L-serine, and it's considered investigational. So that those two I've kind of run into. But there is a list of other experimental or drugs that are in clinical trials that cannot be taken concurrent to the drugs that are in the platform trial, and that you would review with your physician.
Thank you. So Catherine Small and Allison are on the line. There are patient navigators, and I have a question for either one of you, which is from someone who lives in North Virginia, and they want to know if they have to travel to Boston.
Yeah, so I think, if I'll direct you, if you go on our website, you can get my contact information. I'm happy to talk one-on-one via email and kind of work through that. But Really, I think the goal of having 54 partnering sites nationwide is that we're trying to make the trial as accessible as we can locally to folks. So I'm happy to work directly with you to try to find something a bit closer to home. I think that would be ideal, really trying to limit the burden of travel because there would be, 7 or 8 in-person visits over those 6 months. So not super frequent, but not infrequent either.
So we definitely have a site in Virginia, but my geography is that bad. I don't know if it's north or south, but it's in the state. So, but Catherine,
Yeah, the University of Virginia.
Yeah, is available to help and make introductions to if needed. So there's a question about one of your slides, Irfan, and I'm not 100% sure I understand the question, but it was the one where you showed the different doses, and the question is whether these are patients kind of before and after treatment.
Yes, exactly. So what was shown on that slide is the change from baseline to week 12 in people who have a disease called MSA. And that change is on a measure which is sort of like the ALS-FRS-R. It's called the Unified MSA Rating Scale. It's just a way that we use to assess how people with MSA are doing. And so that measure is the change from baseline.
There's also a question about the Parkinson's PET imaging study. The question is, why did the inflammation appear to increase at 8 weeks compared to 4 weeks?
Right. So it's again a great question. I think that, any one patient, that was a very dramatic reduction from the baseline to week 4, and then from week 4 to week 8. I don't know how meaningful a few, sort of couple percentages points are in that one single patient. But when we look at the means across many different patients, it's clearly reduced over time. And it's quite significant and in regions that are of importance in Parkinson's disease. So, this is why we have bigger sample sizes, because there's a lot of variability or noise sometimes in these measures, and the statistical significance across many patients is really what we're looking for.
So another question for you, Irfan, is Verdiperstat offering expanded access, and would this drug be of benefit in combination with some of the other drugs being currently tested?
Right. So I think that I will let maybe the Merit talk about the EAP or someone else. We, in theory, are very supportive of access to people for access to Verdiperstat. The second question was about combinations. I think that this is another great question. The trials are not designed currently to look at combinations of the drugs in the platforms together. But certainly if one of the drugs becomes successful and it becomes available to all patients with ALS, it will be used in combination. And we believe that Verdiperstat is very complementary to many of the other potential medicines that are available for people with ALS or that are in studies for people with ALS.
Yeah, I'll add that in the study, obviously everyone could be on standard of care. So people are allowed to be on riluzole, on Radicava. And so there will be some people that are on combination treatment. They're on those two plus the active treatment. As far as the EAP, so we're very happy to share that IMALS has given a grant for us to do a small EAP with Verdiperstat, and Biohaven's kindly matched that and providing the drug, and that will be at 3 centers, pending FDA and ethics board approval. So the timing we're hoping will be in the spring, but we're working on the protocol, and that'll be at Northwestern, Duke, and Mass General. So there's a question about how do you pick from the list of those interested who can be in the trial, and maybe Jinsy, I'll have you answer that one since you're a site investigator.
Sure. So a lot of the sites, including ours, actually, I'll just speak to our site. Our site is in New York City. We're at Columbia University, and we have definitely had a lot of interest, which is fantastic. Our site really got started in January in terms of enrolling, and we have almost 85 patients who have expressed interest and eligibility. And we've been trying to contact each one individually and keeping them in the loop, but it's been difficult to keep in touch with everybody every week. And we've had to prioritize the list to ensure that people that were getting close to their eligibility time, so people that were going to time out, so they're close to their 3-year cutoff, or if their breathing is very close to the cutoff for the requirement to enroll in the study, we had to prioritize those patients first to get them in so that they have the opportunity to participate in the trial, because they might have only this one time to do it. And then as we continue, we will systematically contact each patient. So all of the patients that have expressed interest, we have room to accommodate, but we have to spread them out so that we can accommodate everybody according to their symptoms, their time of onset. And we do want to make sure everybody's included. And I know that all of the investigators at all of the sites want to do the same. And I think, Mary, you might have touched on this, that we are trying to make sure that we're including more sites even in the future to accommodate the continuing amount of people who are interested, the growing number of people who are interested in participating.
Yeah. We do, fortunately or unfortunately, only have one site in New York. It's a great site, but it's a big city. It's a populated city. But yes, so we are looking at adding a couple other centers in the New York area, as well as other parts of the country. But thank you for being interested, and hopefully it will work out. So there's a question about Verdiperstat mechanism of action. So is Verdiperstat intended to improve the trash removing function of microglia, or is it also hoped that will impact synaptic function and glutamate excitotoxicity as well?
Right. So these are kind of great questions. The myeloperoxidase enzyme, which is what Verdiperstat inhibits, is contained in microglial cells. And one of its key functions is to take certain things like hydrogen peroxide and create very toxic molecules like hypochlorous acid that are very reactive. And so I think its primary mode is focused on oxidative stress. It's not so much on autophagy, which is the trash removal disposal system within the cells. And it's not so much on glutamate excitotoxicity, but all of these kinds of mechanisms converge together. So I think this is what makes the previous question about combinations a really interesting one, is that we hope that by themselves these drugs work, but potentially in combination they can be even more powerful.
Thank you. So Suma, I'm going to ask you this question because of your expertise in research in PLS as well as ALS. But the question is whether this mechanism of action or this drug might have a rationale in PLS.
That's a great question. And the question of whether ALS drugs work in PLS always comes up. And then the discussion surrounding that is PLS part of the same spectrum as ALS, but a slower progressing end of that spectrum of ALS. For Verdiperstat, this has not been tested in PLS before, but neuroinflammation is noted in both ALS and PLS. And we've done imaging studies that have shown that there is inflammation in PLS as well. If there are overlapping mechanisms, there could be a potential, but that needs to be tested in a study.
Thank you. So another mechanism question. Are the glial cell inflammation activation possibly more specific for upper motor neurons than lower, which would suggest the drugs benefit more selective improvements? And I don't know, Irfan, if you want to take that first, or maybe something you could add.
Yeah, so I think that this sort of piggybacks off the previous question that Suma answered. And I think, probably the inflammation and glial cell activation is relevant in both regions. It's easier to see perhaps in the brain on things like PET imaging, but when you look at pathology, there's certainly similarities between what's going on in the brain and in the spinal cord.
I think Suma, I mean, I know you and Nazan did some spinal cord PET PBR28 imaging studies. Do you want to share that a little bit?
Yeah, spinal cord imaging is would give us answers theoretically about inflammation and other mechanisms at the lower motor neuron level. It's not easy to scan the spinal cord because the tissue volumes are very small when compared to the brain. But the study is still ongoing to look at can we quantify that inflammatory changes in the spinal cord in different parts of the spinal cord, the neck, as well as the lower part, the thoracolumbar spinal cord. So that study is ongoing at the moment. We should have some answers shortly.
Thank you. Just for like a little interlude, there's a very nice comment about the majority of women on the Zoom. No offense, Irfan. But.
Well, we know it was Women in Science Day last week. There was a special day, so we support women in science and enjoy collaborating with all the wonderful women on the panel.
Thank you. So there's a couple again, kind of platform related questions. So one is what the FVC cutoff is for the HEALEY trial. Jinsy, if you want to answer that one.
Yeah, I think it's 50. It has to be greater than 50% for the. So that is a number that is traditionally lower than what previous ALS trials have included, but it allows for more flexibility and more inclusion of people living with ALS to have an opportunity to participate in the clinical trial.
Great. And then Suma, there's a question from someone who was just enrolled in regimen B, the Verdiperstat. They're asking, once they complete 24 weeks, how long would Verdiperstat be available in open label extension in case I decide to do that?
Yeah, so currently, once you complete the placebo-controlled portion, you roll over, you have the option to roll over into the open label extension that extends for about a year. Beyond that, there is no further extension that is built into the trial protocol at the moment. And depending on the results of the placebo-controlled portion, future decisions will be made on further extensions or not.
Great, thank you. So there's a question about whether being on AMX 035 is exclusionary from taking part in the trials, and I could answer that one quickly. So AMX 035 is not a FDA-approved treatment for ALS right now. It's considered experimental, and for that reason, it would be exclusionary in the HEALEY ALS Platform Trial, as well as every other ALS trial right now. Should it get approved by the FDA and come on the market like Riluzole and Edaravone, then it would be allowed in all trials unless there was some safety issue. So one of the slides mentioned a non-statistically significant difference. They're asking if we could explain that since and interpretation of the results.
Sure. So there were non-statistically significant trends towards benefit when MSA patients were treated with Verdiperstat over 12 weeks. And so what does that really mean? When we design a study like a phase 3 study or like the HEALEY study, we're generally powering it with a large number of people, a large sample size, so that we can see an effect over, say, 24 weeks. And in the MSA study, it was not a phase 3 study. It was really a phase 2 safety, tolerability, biomarker study. And in those kinds of studies, we call them learning studies. So when you do a learning study, you don't know what you're going to see. You measure things usually for a shorter duration so you can learn about the drug and the potential impacts. And what you're really looking for are there things that make you believe and teach you how you could do a proper phase 3 trial. And that's what the phase 2 trial in multiple system atrophy taught us. We saw these non-significant trends, so we knew statistically how many people do we need to see significance, and designed a trial, a phase 3 trial that's currently ongoing in people with MSA to show a benefit. And so that's a lot of sort of statistical speak, but the simple answer is when you have small learning trials, they're not really supposed to show you statistical significance on clinical outcome measures. They're supposed to help you design the phase 3.
There's a question about someone living outside the U.S. in Peru and whether they can participate in the trial. And I'll maybe answer that one real quickly, that this particular trial does not have an exclusion for international patients. There have been other studies that have done that. However, we do ask that people are able to live close to a center. So for people in South America, I know that some of them have talked to the sites in Miami, Fort Lauderdale, and some people are able to come and stay here for 24 weeks, others can't. So I would just encourage you to reach out to those 2 sites. And again, if you need help making those introductions, Catherine Small here, our patient navigator, whose contact you can find on our website, can help make those introductions. We have another question kind of related again back to the microglia in the spinal cord, and I think it's worth probably going back to that, which is from your presentation, you showed PET images of the brain and how it reduces microglial activity there. Does Verdiperstat also make its way into the spinal cord and reduce activity there?
Right. So a couple of questions in there. The first question is, does Verdiperstat get into the spinal cord? So the answer to that is yes, we believe it gets into the spinal cord. We haven't actually measured the levels of Verdiperstat in the spinal cord, but if it gets into the brain, which we have seen on things like PET imaging, then it probably gets into the spinal cord as well. In terms of what it does in there into the microglial cells, again, this goes back to sort of the same line of questioning before. Suma described these studies that's being done currently to look at PET imaging of the spinal cord to see if there's microglial activation. So those kinds of studies have not been done with Verdiperstat currently, but the brain is sort of a window to what's going on probably in the spinal cord. That's what we believe.
I mean, imaging with PET tracers of the spinal cord is very technologically challenging because of the size and the bone. So that's some of the limitation to looking at it. So there's a question about if someone crosses over into a different regimen, is there a possibility of receiving placebo then, or do they absolutely get the study drug? And I think this question means that, let's say you're the first time you're assigned to placebo, you get through your 24 weeks, you decide to not go into open label, but re-randomize. Could you get placebo again? And I'm happy to answer that because we did get that question a couple of weeks ago, and we asked our statistician, and maybe we could have done it ourselves. But yes, there is a chance. It's a very small chance. It's about 6%, but it's not 0. I do wish we could get rid of placebos, but we're not at that stage in the ALS field yet where we can determine if the drug works without it. So there is a small chance if someone did this twice of both times getting a placebo. But again, we have made the open label extension option for every regimen for people. So there's a question, suppose several of the therapies and AMX035, et cetera, prove safe and beneficial and the FDA approves them. Is there a reason to not be on all of them? Or is there a combination risk? I don't know, Suma, you want to answer that first and maybe then Jinsy?
Absolutely. That's a great question. And when we talk about where ALS drug development is headed, when we have multiple drugs that are approved, can we layer them all, or will there be problems with interactions among some of these medications? So far, riluzole, Radicava, and Dippostat can be layered with no interactions. If and when AMX0035 gets approved, and as far as we know, there's no known interactions with any of these medications either. So as and when there are new drugs that are approved, I think we'll have to pause, look at the drug mechanisms, the side effects, and then assess if there will be side effects or not, and then create a sort of a cocktail of medications, if you will, for ALS patients.
Yeah, I don't have much more. You said it really well, Suma. I think it's just, the reality is that the ALS clinical trial landscape is very much trying to be as flexible as possible, realizing that there will be therapies that come on And once they're FDA approved, they're considered standard of care for that region for the US, right? And so the hope is that clinical trials will allow a patient to be on the background of standard of care, whatever that may be, and allow you to continue to enroll in a clinical trial in the future until we find a cure. But there are important considerations. One is the safety concern, right? So is there a medical condition for the participant for not being on all of those things? And the second is the drug-drug interaction. So not all drugs are really great to take together. And so those are the kind of the considerations you have to take into effect when you're trying to do the background FDA-approved therapy combinations plus a clinical trial. And there's a lot of unknowns.
It'd be a wonderful problem to have, and we all want to get there, just like multiple sclerosis that has 20 drugs. We want to have 20 drugs options. So there's a question from someone who's about to go to open label in 3 weeks, and they're wondering what to expect. I don't know, Jinsy, if you want to answer that one.
Yeah, I'll just take it, and then if anyone else has anything to add. I just had this conversation with a person who's participating at another site, actually in Arizona, and was asking these questions, kind of trying to think through the process. But when you decide to roll over to open label extension, it's a different kind of environment in the sense that there is no placebo, right? So that is what's built into the platform trials. The second is that although you're getting drug and you finished your formal clinical trial participation in the placebo-controlled portion, you still have to comply with the protocol. So that does require you coming to the site, making sure you provide blood samples and other assessments for safety to ensure that everything is going as planned, or if you have any new symptoms, because it's very hard to predict. Even myself, and I've done this with patients in other clinical trials, if you try to guess, and people always say, oh, I'm sure I'm on placebo, I'm sure I'm on treatment. You're not ever 100% sure until you really know. So it's important to be monitored, and when you roll over to know that it may not be the same experience in the way you feel, because now you're getting the drug.
Thank you. And I think for our last question, which is a great question, which is why did we not include PET scans in this study, like you did in the MSA trial? And that could be for anyone to answer. Irfan, you want to start?
Maybe I'll start, and I'll say, obviously every trial is sort of designed with certain objectives. And I think really the objective of the HEALEY trial is to look at clinical benefit. It's not to look at biomarkers. It's not to necessarily learn or only learn. It's really to look for clinical benefit, to accelerate therapies being approved for people with ALS. And, it's always a balance. So, we like to include certain things to continue to learn, because that's always important. But having PET scans would make this unwieldy potentially for a lot of people to participate. And the speed of the platform trial is really one of its major advantages to get drugs approved for people.
Thank you very much. So I want to thank Irfan and Mary and Lila from Biohaven for joining us, and Dr. Andrews and Dr. Babu, the leaders of this regimen, for joining us as well. And all of you, our friends who join us weekly. We will record this if you want to share it with anybody else, and hope to see a lot of you next week. Thank you.
Thank you.
Bye, everybody.